everyone, my name is Delaney Field, and I am doing a presentation for Bio 282 Lab for Conceptual Approach for Biology Majors 2 at Arizona State University for the spring of 2021 semester. I will be doing a comparing of the Designing Vaccines for Artificial Proteins article compared to the primary research article of De Novo Protein Design enables the precise induction of RBS neutralizing antibodies. So first up, my article analysis on my article analysis news article on two sites, both the EPFL News as well as Science Daily. This article discusses how scientists are moving towards producing artificial proteins to correctly instruct the body on how to produce the necessary antibodies better than using non-protective antibodies that cannot be produced by biotechnology. It also went over how proteins are very precisely instructed for specific functions of the body's immune system, which antibodies can be produced, as well as helping to target certain viruses more effectively by again producing and targeting immune systems which focus the antibodies to specific parts of the body. And lastly, expanding functional and structural ranges of proteins through biodesign as well as biotechnology. Primary source was the de novo protein again. So this article was published on May 15th, 2020, so in this past year. In the beginning, they were trying to solve the question of how to create biological functioning proteins. They created a protein design algorithm, which you can see in the picture called Topo Builder, which I will explain more later, which actually designs scaffolds in the proteins for irregular and discontinuous neutralization epitopes, which are also known as antigens inside of the body. The scientists in this research article generated focused ones to help figure out responses to a respiratory syncytial virus fusion protein, also known as RSV, and it was used to help boost site-specific and targeted responses. In the end, they found that the design strategy of computational design involved the topo builder, led to more advancements in defining and building the proteins that can be used for more targeted results again, in leading to new areas explored by biotechnology through an increase in control that this process was able to allow. So in the picture, you can actually see the RSVF where this was actually started. The proteins were taken out and arranged from low to high structural complexity, and then they were put into the computational design, which allowed for 3D assembly. And through this process, they were able to kind of break them up and use a different template for building. They were able to take that to create a synthetic cocktail vaccine, which would be able to focus on those different areas at multi-sites. They used this on mice and other different animals. And then by using those neutralizing proteins, they then were able to create defined antibody specificities, which in this case were able to attack the RSV virus because of those proteins that were able to build up and be more useful and focused. In the conclusion, the article does accurately describe the primary sources and its research and findings. The article does explain new advancements in biotechnological processes, which is emphasized by the research and their findings of using that Topo Builder template building to change the computational designs of proteins based on their initial structures. And by creating these new synthetic vaccines, there can actually be more focused areas when creating vaccines, which can impact the future of the making of vaccines, which we can even see in this past year. This is explained in both the article and primary sources, with the primary source going more in depth. But in the end, the article still accurately represents the primary sources. So therefore, it is a article that can be used to understand the primary source. And lastly, my citations for both my slides, my news article, as well as my primary source. Thank you so much and have a great day.